Okay, good morning. Everything okay with the projects? Yes? Almost, okay. So, the exams are Thursday. We haven't told you the location yet because we haven't found a place still. It's likely, huh?
I think this is in this exam. I've seen it.
Ah, really?
Yeah.
Okay, so maybe they found this. Okay. But yeah, just I forgot to send this email, but now that I'm just now that you're here, I'll just tell you that. So okay, I know each of you have different projects in different topics, but just be aware that you're going to be tested, possibly, on everything that you learned. Okay, the whole. So just because you did a molecular dynamics project doesn't mean that Monte Carlo is off the table, doesn't mean linear algebra is off the table. Just because you did Monte Carlo doesn't mean that molecular dynamics is off the table. Okay, but don't let this stress you out. I'm just letting you know for your information. Okay, so welcome to the next 12 lectures of molecular biophysical chemistry. Okay, so this is an attempt to introduce all of you to the physics of biological systems. Trying to understand or use physical models to understand, because this is an area now that has grown and developed quite a bit in the last 2 decades. Many of you may not necessarily specialize in it, but there's a good general knowledge that you should have in case you go to a seminar or something later on in your careers about the ideas behind molecular biophysical chemistry. So a quick outline of what the course is going to be about. I will put these notes on Dropbox or Google Drive and then share them with all of you. It's going to be updated every week, basically. So you can take it on a weekly basis. So today we're going to spend most of the time with a sort of a general motivation just to get you guys thinking about problems in biophysics. And because, if you're like myself, came from a background in physics or math, there's a lot of nomenclature, there's a lot of language and ideas that you may not be familiar with. So I want to just get you warmed up to some of those ideas that, or questions that we may ask, that a physicist may ask to understand a biological problem. And one of the things we're going to spend quite a bit of time on for maybe 2 or 3 lectures is getting a sense of length scales, time scales, and energy scales associated with biological systems. So because if you're a physicist, those are the kind of 3 things that you would want to think about to get an intuitive sense of what are the processes that control the system. Length scales, time scales, and energy scales. Then there's going to be, it's not particularly exciting to learn about, but you have to learn it. Learning about structural molecular biology, so DNA, RNA, proteins. We're then going to spend quite a bit of time working on simple lattice models that allow you to understand processes in biology. Protein folding, protein dynamics, drugs that may bind or interact with proteins. And then, so all the discussions we're going to be talking about are going to be related to biological matter in the cell. And the cell is made up of a lot of liquid, water. And so we're going to spend a lot of time trying to understand how to model or think about interactions, molecular interactions between water and biological systems. Okay? And then finally, probably towards the end of the course, we will begin to think about how tools like Monte Carlo, molecular dynamics, electronic structure, how they can be used and applied to understand biological systems. So this class is going to be very, I hope, again, very interactive. Please stop and ask questions whenever you want, okay? Okay, yeah, and so there's, how many of you know about Poisson-Boltzmann, Debye-Huckel theory? Okay, basically, these are mean field type models that allow you to understand if I have a charged surface, how does the potential created by the ions, by the charged surface, change as a function of distance from the charged surface, okay? So, so these are very simple mean field models, and we're going to discuss them in the class. Okay, so a couple of references for you. The book I really like is this one on the left here, Physical Biology of the Cell. It's written by a physicist, Rob Phillips, and they have it on reserve in the library. And Rob Phillips goes through many of the things we're going to be talking about today, and he introduces biological concepts for physicists, so it's quite nice. So I highly recommend you taking a look at that book. The one in the middle is more biological, so it's introducing you to concepts of how to think about soft matter interactions. And as you will see, if you want to do molecular biophysics, you want to understand molecular biophysical interactions, one of the things you also need is physical chemistry. And so a textbook that I highly recommend is Physical Chemistry by Atkins. And I'm going to, well, cover many of the topics in these three textbooks. The structure of this course in terms of the grading is the following. You'll have a final exam. There will be a couple of exercises, or homework assignments. And then in the past, we've had usually 2 days of presentations on topics related to biophysics. So you choose, if you want, I can suggest topics to you to talk about. So for example, last year, someone was interested in DNA computing. So, you know, there is and there was, well, more like there was an area where people thought they could use DNA to do classical computing, DNA molecules. Okay, so there are topics like that that are at the boundary between biology and physics, and so each of you will give a short 10-minute presentation on and discuss those topics. Okay, so let's get started. So what is biophysical chemistry? So the thing I like about biophysical chemistry is very interdisciplinary. And so what we do in biophysical chemistry is we work at the boundary between biology, physics, and chemistry. And in each of those lines on the triangle, there's individual fields that have developed. So biology and physics is biological physics, physics, chemistry, there's physical chemistry, and then between biology and chemistry, there's biochemistry. And these are actually individual branches of science. And what we're going to try and do in the course is expose you to some important ideas across all these different things. So you have, you've developed a lot of knowledge in physics. I'm going to take you back to high school and remind you of things that you learned in chemistry that are going to be important in understanding biological problems. Okay? So when I tell you, when I ask you, if I ask you, what type of things do you think about in soft matter biophysics? What comes to your mind? What types of systems are we thinking about? Let me try and paraphrase. So let's take a cell, biological cell. What's inside the cell? What characterizes the cell? The nucleus. What's in the nucleus? DNA. Okay. What else? Good. Yes, that's true. DNA, RNA, that's a good one.
Lots of proteins that binds them.
Lots of proteins that bind them. Yes, there's lots of proteins in the cell. Just out of curiosity, how many proteins do you think there are in a cell? Just a guess. Huh? Yeah, millions probably, maybe billions. Billions of proteins in a tiny cell of 1 picogram. So the weight of a cell, E. coli cell, is 1 picogram. 1 times 10 to the minus 12 grams. So in that object there are millions and billions of proteins. What else? What else is in the, so we talked about nucleus, DNA, RNA, proteins, mitochondria, cytoplasm, neurons. Neuron is a type of cell. Neuron is a type of cell. Golgi apparatus, there you go. So now he's remembering all his biology. Fats, lipids, water. How much water do you think there is in the cell?
70%.
But true. 70% of the weight of the cell will be water. But how many water molecules do you think there would be? Just guess.
Okay, so picogram is the weight of
Yeah.
the single cell. Pico means
10 minus 12.
minus 12 into 0.7. That's
10 to the? 10 to the minus 13. No. If anything, it has to be a large. It has to be a large. He said millions of proteins. So it must be several of orders of magnitude larger than the number of water molecules. So now, why?
Oh, so these were grams.
Yeah. So not the weight, but the So you have to do. You have,
about 10, almost 10 to about 10.
yeah, 10 to, about roughly 10 to 10. Exactly. And so we're going to go through, probably not in this lecture, but the next lecture, getting a sense of these numbers. Getting a sense of these numbers, getting a sense of these length scales, because now, if I know that there are 10 to the 6 or 10 to the 7 proteins in a bath of 10 to the 10 water molecules at the length scale of micrometers, I can ask and think about questions of how concentrated is my cell, and if I was to think about a particle undergoing a Brownian random walk in this crowded environment, would a Brownian dynamics be sufficient to describe the diffusion of the particle, or would being in a very crowded environment with all these different things affect the way that the particle moves around? Okay, so that's where we're going to go. But in order to get there, we need to build our intuition on these various concepts of length scales, time scales, and energy scales. Okay? Okay, so you guys have answered a lot of these questions already. So there's your cell. I have a pointer. Do you know if this thing has a pointer? Or
I don't think
It's
so.
not.
There's a stick.
where's the stick? Here. Okay. Okay, let's do it old school. All right. So there's your cell. Yeah. Mitochondria. Well, we forgot about the cell membrane. So the cell is confined in the boundary of it is the cell membrane. The cell membrane is what allows stuff to go in and out. Okay? So when I undergo cell division, so let's just think about this. Cell division takes, for an E. coli cell, So the time it takes for a new cell to be created takes about 3,000 seconds, roughly. Okay? And so now, if you want to generate a new cell, you have to generate millions of proteins on a time scale of 300 seconds, and you also have to expand the cell to reach a population of about 10 to the 10 water molecules in 3,000 seconds. So, if you do the math, you'll see that this is insanely optimal. It's very quick, it's very efficient, it's very fast. So, how and why is it so fast? Those are the types of questions we want to ask, and we're going to try and answer. Okay? Okay, so that's the cell with all the organic soup in it. This is your double helix DNA, made up of DNA bases. And then there are all these proteins. Okay, well, all these and many, 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 many more. And basically, proteins are the building blocks of functionality in biology. So all function, most function in biology happens because of proteins. So for example, the reason you're able to see right now with your eyes is because light is interacting in an optical way with some proteins, which is causing mechanical changes happening at the back of your retina, which then triggers off a whole set of processes that allow you to see. Okay? Muscle contraction when you walk is all because of the movements and motions of proteins. Okay? When you breathe in oxygen, oxygen is taken up by a protein called myoglobin, transports it because of a protein. So there are numerous examples. Your sleep, the reason you want to sleep in the night, if you're an average person, and wake up during the day is because of something known as circadian rhythms, which are because of proteins. So the brain is expressing certain proteins in higher concentration during the day, reduced ones in the night, and this is what controls your sleep cycle. Okay, so proteins are at the heart of every possible thing that you can imagine. And so this is a type of system that's rich in a lot of physical properties. And so that's why both physicists and chemists are very interested in trying to understand the molecular processes associated with all these types of proteins. So just to show you here, we're going to go into the details, but these are, how proteins look like at a nano length scale. So, the length scales you want to keep in mind here are roughly on the order of 10 to 30, 40 angstroms. Okay, so 10 times 1 to 4 nanometers. Okay, that's the typical length scale of small globular proteins. What else? Okay, so we talked about lipids. So these are fats, for example, that form the surface of the cell, so the cell membrane. And these cell membranes are porous. And the reason they're porous is because they allow for ions to diffuse in and out of the cell. These are things that cause action potentials that then are at the heart of why neuron cells function. Okay? And of course, there's a lot of water. Okay, so just to build your... Is there a question before I move on to some things to make you hungry? Okay, so how many of you like pasta? So. You don't like pasta? So you're living in the wrong place. How many of you make pasta? Okay. When you boil pasta, you notice that you form this white froth on the surface. What is it? What is it?
Some kind of fat.
Fat.
Some kind of protein, I can't say.
I don't think it's fat. Really, the manufacturer procedure of those put some cake.
They put chemicals inside. Okay, and
that is all I usually think.
We don't say CC.
I see. It's not that. It's not extra chemicals. It is a chemical. It is a chemical, but it's certainly not an extra chemical that the manufacturers put into the system. Okay. So what makes pasta? What's the ingredient?
It's wheat.
Flour and egg. Egg. Maybe the white of egg that is coming.
Really?
Don't they put to beat it, the flour, to make the dough?
Right. So okay. The flour is the main ingredient. Then you put things like egg to change the consistency and all this stuff. But the main ingredient in pasta is starch. And starch is
Sugar.
carbohydrate.
Yes.
It's a sugar. Okay. And do you remember what sugar looks like? At the molecular?
No, no.
By the way, it's not only white, it can also be brown. But at the molecular scale, how does it look? What is it?
C6H.
For
6.
Aha, good. And so that's made up of OH groups,
Yes.
hydroxyl groups.
Yes.
Okay. So when you boil pasta, okay, the pasta molecules have more kinetic energy, okay, and so they,
Pasta molecules.
yeah. When you dump pasta into, why does it change its form? Why does it go from hard to soft?
Because we
Yeah,
can
such an energy from the
the molecular value.
You change the molecular, okay. And so the reason it's able to do that is because when you put it in water, you give them enough thermal energy to break. And when it breaks, the starch molecules go flying out into the liquid, and then they migrate to the top of the surface. Now, is there an energetic penalty to put glucose or sugar at the surface of water? So, I have this.
Yes, I think it's heavier than water. Sugar is much heavier than water.
Sugar is heavier than water.
Like C6H12O6.
So, oh, you mean compared to an individual water molecule?
Yeah.
Okay, but but then why does it go to the surface? Why do you see it at the surface? So let me. So I have sugar. You all agree that it loves water, right? It loves water.
Which kind of sugar we must use?
Take whatever sugar you want. Take glucose, fructose, whatever you want. It's all they all for our purposes love water. And so this is air. And so my pasta here breaks up, and then I have the sugar molecules swimming in solution. Okay, if I take this to the surface, what's the difference in the dielectric constant between here and here?
What's one?
The difference in the dielectric constant between this and this.
1 minus.
The dielectric constant goes from something like 80 to something very low. Because this is I've got water here and I've got air here essentially. So I have, there's a gradient in the dielectric constant. Okay? And so the dielectric constant goes from about 80 to some low value at the interface. So there's an energetic penalty for me to take, let's say, a charge. Let's imagine a charge. If I take a positive test charge and I move it to the surface, there will be an energetic penalty because I have to pay an energy of bad interactions or weak interactions with water to put a charge at the interface. Is that clear or no? No. Okay. Where do charges? Let's take. If I tell you sodium chloride, you mix salt in water. Okay. Do the charges want to be? Do they maintain the crystal or they dissolve? They dissolve, right? So they dissolve because, well, there's an entropic factor that drives that, but when my sodium and chloride ions are separated in a bath of water, they have strong charge dipole interactions. Right? Sodium plus, chloride minus. I can think of water as a dipole. They're favorable charge dipole interactions. Now, let's take that test charge, sodium, and put it at the interface. Is it going to be as favorable energetically for the charge to be at the surface versus being deep in the liquid? No. Why?
I think its entropy is like more of an answer here, because why would it go and stay at the surface? It would generally mix everywhere to increase entropy.
Okay, but it does go to the surface. So it's not just entropy,
Yes.
and this is the point. So when I go to the surface, I have an energetic cost to put it there. But how do I compensate for that energetic cost? Stick together. So if I stick together, I form stable interactions between the molecules. Okay, so you'll see this in the next slide with where I have more pictures here. So, okay. So the main ingredient is carbohydrates. This is starch. This is glucose. We all agree that these guys like to interact with the water, the liquid. No doubt there. Okay, this should be very clear. But now if I put this at the surface, up here, it's going to cost energy. I'm going to lose favorable interactions of the starch molecules with the water. And so the reason why things aggregate, the reason why you see the white froth, is because now lots of these molecules try to form something like a precipitate. They interact strongly with each other. So they compensate for their loss in interactions with the water by forming interactions between themselves. And so the froth that you see at the surface is because of starch molecules interacting with each other.
But like in a more fundamental level,
Yeah.
they could have stayed down and interacted with water, which is generally more favorable.
Yes.
But why do they go at the top, which is unfavorable, and then regroup and work against the more logical?
Okay. This is a very good question. There is another driving force that pulls the starch molecules to the surface, and it has to do with something with polarization. So there's an enhanced polarization that the starch molecules and the water around it feel when they're at an interface. But I don't want to get into that now. Okay, but we will get into that later on in the course. But what I want you to, so the purpose of this exercise is the following. There are some very basic problems in your day-to-day life that involve rather simple physical chemistry. The thermodynamics of aggregation, the thermodynamics of nucleation in the bulk versus the thermodynamics of aggregation at the interface. And the other thing you should pay attention to is that the concentration, of course, of those things at the surface is much less than what's going on in the bulk. So it It's not that the aggregation, it's not that it's not happening in the bulk. It does happen, but very rarely because of entropy. But the point is, the lower concentration of molecules that get to the interface then have to pay a price. And in order to compensate for the loss of interactions with the environment, they form the froth here, which is just an aggregate of starch molecules. Okay, yeah, please.
Why not? Okay, like if you have your pasta.
Yes.
I've understood that the starch goes on top.
Yes.
It breaks away from the pasta.
Yeah.
The pasta strands.
Yes.
Does it break away does it break off as starch or as separate like?
It depends on the temperature. So if you have high enough temperature to break covalent bonds, then it will break away as individual molecules. But
When do we expect such a?
so in this case, you will have objects that look like this, like dendrimers. So you have strands. The strands along the starch will break up as chains, rather than separating out into individual groups.
And then if we understand it in terms of charges,
Yes.
I expect that if same charges are maybe same charges not like to be together. I mean,
What do you mean by which?
if
who's-
I'm to understand it in terms of charges, because
Yes.
I explain it
So-
in terms of
Okay, so okay, we're getting ahead of ourselves, but so this object here, the OH group, is a dipole
yes.
because I have a negative charge on oxygen and a positive charge on hydrogen. It's a dipole, and the way you should think about the interactions of the starch with the other starch is it's dipole-dipole interactions. Okay. Okay, so. Number 2. Why are different fruits colored differently?
Because
The nutrients.
Because of the nutrients.
Composition.
Okay, but why?
Absorption and some colors are absorbed. some
Yes.
are-
And
I
they,
think it's protein that does it.
protein?
Fruits don't have protein.
No. No, not protein. Not in these guys.
Because I know one protein, carotene, in
Yes,
eyes, that
yes.
is
Is it carrot?
and it comes from carrot, so I was trying
Ah.
to understand.
Yeah, no, that's a good connection. But the molecule itself is not a protein. So carotene is not a protein. Okay, but the protein.
Carotenoids, something like that.
Yeah, but it's just a. So the principle is right. It's some object, protein or molecule, whatever. It's some molecule, different molecules in different fruits and vegetables that control the energy of absorption. Okay, and depending on the energy which they absorb, they will absorb some colors, but allow other colors to go through.
So I think if let's say if we look at mango, then
Yeah.
initially it will be green. As time goes on, it
Yeah.
changes its color.
Yeah.
So
Yeah.
there must
Yeah.
be
Yeah.
something like more concentration of something.
Absolutely. Absolutely.
And,
Yes. Yeah. So the ripening process is fundamentally related to changes in morphology and chemical composition.
There is also one extra approach where you put acetylene to ripen it. An extra. They do it before time.
You do what?
They put acetylene to
Artificial.
ripen.
Yeah. Sure. Yeah. Although that,
C6 has to-
but that changes mechanical properties. Okay, and then of course that's coupled then to optics. So the acetylene doesn't directly change the optical properties. The optical properties change because probably the whole morphology changes when you add an impurity.
Okay, so I'm just making an blind
Yeah,
connection.
please.
Let's suppose like when mango is green, it is
Yeah.
sour. When it is yellow, it is sweet. So more sugar, so maybe more stars. That maybe.
Well, in fruits, it's mostly fructose,
So,
not glucose.
so I need some kind of sugar.
Yeah.
The concentration
Maybe.
is rising, so
Yeah.
it's correlated with the color. The
Probably. Yeah, I don't know actually. I haven't thought about this one. But yes, for sure it must be related. For
What
sure.
about the tomato? Color in the skin.
What about?
What the tomato?
tomato also gets
fructose?
no? It doesn't have to be fructose. I
So
mean
he's saying some type of sugar.
the,
No, I thought that you were giving something universal, maybe.
no, I'm
No,
saying
no.
taste changes. Like tomato is
Yeah.
not supposed to be sweet, but yeah, a very ripe
Sure.
tomato may be sweet.
Sure. No. It's true. It's true. I mean, I
What I'm
think.
saying is the taste changes with time.
Yes.
And also its optical properties.
Yeah.
So I was just trying to correlate.
Make a no, and this it's a very fair observation, and although all I'm saying is I don't know actually because I haven't thought about that connection between the change in the taste and the change in the optics, but for sure there must be. So all of these guys have different. There's your keratin. Yeah? And the different colors of fruits are related to the optical properties of different types of molecules. And these different types of molecules absorb light at different frequencies. And so the reason why you see tomato as red is because it has a high concentration of lycopene, okay, and it transmits only red light after optical activity. And so one of the things that we're going to go towards is, how can you use simple physical models, like a particle in a box, to understand these changes in optical properties. Again, you'll see that you can in fact do that. Again, you can learn a lot about a system from these types of simple models. But what I want to emphasize is that it's also important to have a strong understanding of more fundamental molecular interactions because they play also an important role. Yeah, please.
You realize that some fruits have a different color on the surface and then inside they have a different color. So where is maybe for example the carotene? Well, are you talking of the carotene? Because the carotene leads to yellow color.
Or carrot, so.
But we've seen mangoes that are red.
Sure, a very good question. And so you can generate different types of carotene with different lengths. Okay? And depending on the length of the carotene, if you think about your particle in the box, the length over which the electron can delocalize will change the excitation energy, the absorption. So, absolutely. Variations in color will be strongly linked to differences in not only the length but also the morphology, the environment, all these other things. Okay, so that just restates what I said. Okay, so now you all live in Italy and you're big fans of cappuccino or not, maybe. What makes a good cappuccino? Okay, this is not a scientific question. What makes a good cappuccino?
Cheap price.
I see. Okay, so I'll tell you what makes a good cappuccino for me. A good cappuccino for me is something that has a nice, thick foam. The foam is the most important thing.
Why?
Why? Because it's so before you suck the hot liquid, you get a soft feeling. So this is very important, and it looks good. Okay, but now, so that's the scientific part. What's the foam made up of? It's milk. It's maltose is the sugar.
Lactose.
No,
Lactose.
Yes. Lactose.
That's certainly one of the things in sugar. I mean, in milk. What else is in milk? Very good. Calcium, definitely. What else? Why do kids drink milk? Okay, and it
Iron.
amino proteins. Okay, there are also fats in that, but there are proteins in milk. This is sort of one of the calcium and proteins are one of the essential things. That's okay. So that's the milk. The foam comes from air bubbles. They're air bubbles, and air bubbles interacting with milk proteins. So there's the thermodynamics of the creation of something known as a micelle. Yeah, I'll show you what that is shortly. But I have air bubbles that I've created because I've taken the thing to the boiling point. There are bubbles forming, and now bubbles have milk proteins, and these milk proteins have a rather peculiar shape. So one of the proteins in milk is called casein, and casein has, it looks like a tadpole, so it has a head that likes water, and it has a tail that hates water. And when I say like and hate, I mean in terms of energetics. So if I was to look at the enthalpy of the interaction between this and water, it would be negative. This and water would be positive. So when I have an air bubble and I put it in contact with an object like this, you form structures known as micelles. So the reason it forms this is because there's water all around here. Does the water want to interact unfavorably with the fat? This is the oily part? No. So this is exactly related to when I'll show you later. But so this is the oily part. It doesn't like water. The tail. This is the part that likes water. And in the middle here is essentially air. All right, for all practical words, you can think of it as a vacuum, because the concentration of air molecules is very low. And so the most thermodynamically stable state to mix this with water is to form this. And so your foam that you see is at the molecular scales made up of these objects. Now, how many of you do your laundry? How many of you wash your clothes? I hope so. When you wash your clothes, do you wash your clothes with water, or do you wash your clothes with water and something? What do you put? Detergent. Why?
Because it reduces the surface.
Reduces the surface.
Surface tension and so on. The way
These are certainly all related to surface tension, that's true. But I think Joe, you said, so the detergent,
I said to dissolve.
dissolve the
That's
dirt, dissolve typically the oily things. And detergents are made up of things that look like this. And so the reason why detergents work The reason why you're able to pull them out, the dirt off the clothes, is because now,
The dirt stays inside the. So
exactly, that comes in, sticks it, boom, and flushes it out. That's why if you wash your clothes with water, it's not going to get rid of stains. Okay, so again, a very practical example of, you may not be realizing or thinking about it, but there's all this interesting stuff going on at the molecular scale with pulling off dirt from your clothes. Okay? So one last thing. Okay, this is a bit of a, well, might be a weird example, but anyway. So, okay, if you look at your teeth, you know, most of us like when we're born, we have nice white teeth, well, for the most part, and then as you start to drink coffee or you smoke or whatever, you don't brush your teeth, they get stained. And the reason they get stained is, so your teeth are made up of a mineral, it's called hydroxyapatite, which is a mixture of phosphorus, oxygen, and calcium. It's a very hard material. And on the surface of the tooth, you get molecules like caffeine, chocolate, that both form very strong interactions with the surface. And so, what you do with a toothbrush or what you do with a stain remover is designing something that will pull off the stain from the tooth. Okay, so yet another example of a very practical example of where biology meets physics meets chemistry. And what we're going to be trying to do is now dissecting this at a more physical level. Okay? And so I'm going to revisit a lot of these examples, but with much more clarity in what we're discussing. Okay, so now I want to give you, so as I told you, this first lecture is more general, more motivation, just to calibrate yourselves on the things you're going to learn. I want to give you now some more, let's say, real examples in biology, where molecular interactions play an important role. So there are these fish and insects that are able to survive in very cold conditions, sub-zero temperature. The question is why? Why are they able to do that? And so there are people who spend their whole career trying to figure out the thermodynamics and kinetics of things known as antifreeze proteins. Actually, there's a person at Ohio University who's an expert in antifreeze proteins. But so how does this work? Okay, how can you get an insect that's able to survive at minus 15, minus 20 degrees? So there are things known as antifreeze proteins. Okay, so going back to proteins again. And what these antifreeze proteins look like, so they look like this, and they are able to stick to the ice lattice, the surface of ice, and they can disrupt the thermodynamics and the kinetics of ice growth. Okay, so if you have an ice crystal nucleating, these molecules can bind to the surface and prevent the ice surface from growing. Okay, and so people are interested in trying to understand how these things work, because it's the same, the physical properties of antifreeze proteins, the same stuff of the things you put into your car to prevent the liquid in your car from freezing. I forget the name of the technical term, but Okay, so it's the same. It's not proteins, but it's different. I forget what they're called. Antifreeze agents. Okay. Anyway, it's not important. So we talked about, or I mentioned to you, the membrane, the surface of the cell, and on the surface of the cell there are membrane proteins. Proteins that allow their pipes, nanopipes, and the pipes allow for water to go in and out of the cell. And so I think 2 decades ago, this guy, Peter Agre, got the Nobel Prize in chemistry, or in medicine, one of the two, for the discovery of this protein. And what he did was he did 2 sets of experiments. He took a cell with this membrane protein, he put it in water, and because of something known as osmotic pressure, the thing inflated because the chemical potential of the water outside and inside was different, and so in order to establish equilibrium, there was osmosis. And then he took a cell without aquaporin, so the biologists are able to do mutations and get rid of the specific protein, and the cell remained the same. And so this was his discovery that you need these pipes to allow for water to diffuse in and out of the cell. And so I just show you, this is it's not a movie made by me. Did I show you this already? No, I don't know. Anyway, whoops. Maybe I did, but so this is a molecular dynamics simulation, so a trajectory, positions and momenta as a function of time, classical, canonical, ensemble, position, momenta, independent. Okay. You'll be tested on Thursday. This is the protein. These are water molecules, and they're trying to squeeze through this pipe. It's a small pipe. And what's interesting is right at the middle of this channel, there's some evolution has created a positive charge on the protein. What this means is that it costs a lot of energy to move something like a sodium ion or a potassium ion, so a positive charge from inside the cell, from outside the cell into the cell. So this is a very specific water channel. So it only allows water to go through. Okay? And one of the things we're going to come to is trying to understand how fast does the water go through this channel. And if it was just, does it follow the rules of a random walk versus something that's more directed? So related to this is, so those are water channels. Now I'm talking about ion channels. So there are different channels that allow for potassium ions to go through or sodium ions to go through. And so, for example, in this case, what this picture is telling you is that the geometry, the environment, where the positive charge will interact with some nearby dipoles, has to be just correct to allow for only a potassium ion to go through. If you try to bring a sodium ion, it would not be, there would be an energetic penalty to put a sodium ion there because you have essentially a defect. So there are attractive interactions, but it's not as favorable as putting a potassium ion. So geometry plays and conformation plays a very, very big role in these types of systems because they're soft matter systems. So in soft matter systems, this is what makes them very, very challenging, but also very interesting, is you have a lot of conformational flexibility and on different time scales, from femtosecond all the way to the age of your life. Okay? And so what we wanted to do is to get some tools on how to think about these types of fluctuations. Okay, so perhaps one last more specific example. This is a virus. It looks like a spaceship. Right? It's not a spaceship. It has a head, it has the body, and it has feet. Just to give you a sense, so this is called the capsid. The length scale of this is 65 nanometers by about 100 nanometers. Okay, 120, 130. And the virus has to pack DNA, the double-stranded DNA, into this space here. And it has to pack 10 micrometers of DNA into a region with very tiny volume. How does it do that? How does it do that knowing that DNA, as you know, is negatively charged because of the phosphate backbone? Okay, so the DNA, we will learn about this in greater detail, but the DNA is negatively charged. And that's why, because of the phosphate groups. And that's why DNA is typically complexed with positive ions. So things like magnesium or potassium. But, you know, this is a very interesting packing problem. It's a packing problem coupled to how you screen repulsive electrostatic interactions in a confined environment. Okay? And so the types of models that I was telling you at the beginning of the class, like Poisson-Boltzmann theory, Debye-Huckel theory, are mean field models that allow you to rationalize how you can achieve this type of packing. Okay? Okay, so there's this, let's say, theoretical side of things, of building some ideas of the physical models, but that's on one side. On the other side, we're also interested in getting a feeling of how do I take those physical concepts and translate that into something that I can simulate, where I can do some numerics to understand the behavior, the statistical properties of a protein. And so, well, what are the things that I need to think about if I want to model, if I wanted to develop a numerical recipe to study the NVT ensemble associated with a biological polymer? What are the things that I would need to think and worry about? Well, let's actually back up. Let's forget about proteins. Let's just think about simulating soft matter systems for your, like we discussed in the molecular dynamics. Scusa. So what are the things that go into the numerical recipe if I want to simulate a soft matter system with molecular dynamics? Joel?
On one protein.
That's one point. So I have a possibly crowded environment. So I have many proteins, and the proteins feel forces from everything around. Okay, so it's a many particle problem. Great. So that's a challenge, and I have to figure out how to do that. I need to figure out those interactions. The interactions are one of the most difficult things to get, because how do you treat those interactions? Okay, so you could say, well, I could treat my particles as spherical balls, put in some harmonic or even anharmonic potentials between them, and couple those spherical particles to a bath. They feel some friction in the bath, and one can do that. Okay? And we're going to, and that's one school of thought. That's what people typically do in statistical physics approaches to studying biological systems. We're going to review those things, but then tell you more about what those types of models are missing. What information do you lose when you treat biological systems in a coarse-grained manner? Okay, so this is just, what we just discussed is coarse-grained. Okay? But, okay, so we talked about this, level of atomistic detail, cryo environment, and of course, there's a question of time scales. So every system that I do, whether it's liquid argon, whether it's a water cluster, whether it's whatever you want, will have some characteristic time scales associated with the fluctuations. And if I want to get control over the convergence of a particular property, then I need to know on what time scales do these properties relax. Okay, because that will give me an error bar on the property that I'm interested in computing. Okay, so this is just repeating what we just discussed. So I can take this approach, spherical balls, harmonic potentials, and then try to see what that type of model gives me. What my job in this course is to give you an appreciation for it is that there's much more complexity to treating biological systems just as spherical balls, because they have shape, they have different chemistries, and those different shapes and chemistries interact with the environment in highly non-trivial ways. Okay? And so one of the big challenges, for example, is how do you understand the nature of the interactions between the liquid and the polymer? You know, so let's go back to perhaps a language that you're more familiar with. How do you define the Hamiltonian of this type of a system? What Hamiltonian do you use?
Usually depends on how we model it. If we do the spherical balls,
Yeah.
then maybe you'll just consider the harmonic interaction
Yes.
and potential, also the kinetic energy of that
Yes.
as it vibrates around the center of
Yes. So if you could model your system as a bunch of harmonic oscillators, easy. The problem is these are soft matter systems that are highly anharmonic. First point. Second point, polarization plays a big role here. So treating all these particles with point charges, like we did in some of the exercises in the numerical 2 class, can work, but it may not work for the right reasons. Okay, so polarization is a significant contributor in the interaction of the polymer or the biological matter with the environment. So that's something else that we need to think about. Okay, so I think that the picture is clear. This is something that we all do. People who try to work on biological systems, is when we try to compare with the biological environment, we often work on one protein in a bath, and we imagine that the functioning is of one protein in the back, but actually it's a very crowded environment. And so crowding does have a big role on the functionality, because the proteins don't work independently. They work in a coupled way. So, okay, good. So, let's get started now with a review of some basics. So, this is just to remind ourselves. So, what are the, you all took Biology 101, or, no, not Biology 101. High school biology. Biology 101 is what you take in the US as an undergrad. Okay, so what makes something living? What are the grows? Okay. Respiration, what is respiration at a more basic level? It's energy conversion.
Breaking the food.
Breaking the food down. You take chemical energy, you do something to it, and then you expend it in maybe any other chemical or mechanical energy, So this is energy usage or transformation. What else? You said heat, no. Reproduction, okay? So reproduction. Local motion. I think that's in growth. Yeah. Local, movement, growth.
There's one of excretion.
Really?
Yeah. I think so.
That's the criteria for something to be living, is to excrete? This is news to me. But wait. So, what's something?
Energy usage.
Energy usage.
Energy usage.
Yeah, so no. What's the difference between you and the rock? Is there a difference between you and the rock?
You excrete.
Apart from excretion. If someone needs to use the toilet, they're welcome to go. But, what do you make? What's our building block?
The what?
What's the building blocks? What have we been talking about? Cells. Cells. So, one of the criteria is that I must have cell, and this could be uni or multicellular.
You talked of cells of crystal and so maybe units.
No, come on. Don't joke with me. You really think that the cell that you were talking about now, with organic matter, with protein, is the unit cell? No.
Between the row and then you said cells.
Okay, so I think you're stretching the analogy a bit too far. So a unit cell is just a concept in solid state physics. It doesn't have a subject as the unit cell has life or something, right? So two completely different things. So, what else do we do over time?
We age.
We age.
Excuse me.
Yeah.
Can growth and reproduction be put in the same thing, because in a unicellular thing it's kind of the same?
Well, I think they're distinct concepts, right? Because you grow
Then you die.
individually, but reproduction is creating new life, new cells. So you can. There's a lot of overlap. Yeah.
Living things die.
Living things die. That's an interesting one. That's an interesting one. Yes, true. But I could think of that as number 2, right? I guess
Yeah.
too. Energy. Inability to consume and transform energy from one form to the other. What's happened to us over millions of years?
Evolution?
Adaptation, adaptability. That's at least adaptability. And I suppose adaptability can be put in the same category as response to stimuli, right? So sometimes they put this as separate things, but so they're okay. Now we can have a philosophical conversation about this, which I'm going to try and avoid. But yeah,
should growth be there? I mean, movement. I mean, was living rocks can grow by heating and expansion.
true. But so let's say the way the biologists define life is a combination of all these things. Okay, so it's a very good question, because if you use these definitions, okay, for life, then there's a problem, because then if you take a virus needs to rely on someone else's DNA to do stuff, so it doesn't reproduce. Is it living or not? That's what I would say. Okay, but there's actually people argue about this. Okay, well, what defines the boundary between what is living and what is not? But more or less, this is what these are sort of the properties that define living versus non-living. And what we're going to try and do is visit these criteria at the nanoscale. So what is it that's going on at the nanoscale, at the length scale of nanometers, that allow for efficient processes in each of these things? What are the length scales and time scales associated with this? What are the energy scales associated with energy use? What are the length scales associated with response to stimuli? Okay? So, and all of these involve sometimes mechanical energy, chemical energy, electronic energy, like for the vision. And so those are the things that we're going to try and review. So, yeah, okay. So there's an interesting article in BBC about. So I just told you this, and then this contradicts what I just told you. Okay, so there are over 100 definitions for life, and all are wrong. Okay, so you can go read it if you want. And one of the reasons why they talk about it is because it becomes blurry for things like viruses, okay, to use these types of definitions. Just so we're almost at the end, so I'll end on an easy note. And to me this is very interesting, actually from a deep scientific point of view, is how, so okay, one can define these biological processes with life. What is the definition of consciousness, and can one develop a theory or a concept of consciousness that's built on molecular interactions? So you've obviously heard of these experiments where they take a monkey, they put it in front of a mirror, and the monkey is self-aware. It's able to identify that, hey, I am the monkey. I don't know if it says that. Okay, but they do the equivalent of what we would do. But if you think about a mouse or a bee, does it have the same sense of self-awareness? And is that, so first question, is that related to consciousness? I don't know. And can we describe consciousness in terms of networks of molecular interactions involving all these things? Maybe that's all it is. Okay? And so there are some people who actually are thinking about these types of things. And so you probably know this, there are these things called the happy or moral molecules. So serotonin is a molecule that's released when you exercise or when you eat chocolate, for example. It makes you feel good, or when you feel like you're in love, even this one. Okay? So anytime you think you're in love, it's because there are lots of these molecules being excreted from your brain. And well, not only, but other things, these couple with proteins, they change networks, they change pathways, and you get a certain feeling, a certain emotion. Okay. Can one describe all these things in the language of molecular interactions? Actually, there was an interesting book published that did a study on the levels of oxytocin in criminals, people who were in jail. And it turned out that there was a statistically significant difference between the concentration of oxytocin in criminals versus people who were not criminals. So it's curious. And so this is a very open, broad question on how does one think about non-tangible things like consciousness in the language of molecular interactions. Okay, good. So we have maybe 5 minutes. I'll just introduce it to you and then we'll continue on Thursday. So we're going to spend the next class using the E. coli as, like the hydrogen atom of physics. So the E. coli is going to be our hydrogen atom. We're going to spend a lot of time getting a sense of various length, time, and energy scales in the bacteria. That's how E. coli looks. So E. coli has this, it has this cylinder-like shape and has hair, which allows it to swim. Okay, and the types of things that we're going to be interested in is, well, what is the volume of the E. coli? What is its mass? How many proteins are there inside it? How many water molecules? How many fats? How many DNA? How many mitochondria? And what are the typical energy and force scales that are relevant for processes inside the cell? Okay? And so these are just different images. So the thing that you want to remember is the following. So I think these are electron microscopy. Okay? But this is more or less what we need to remember. So that the E. coli bacteria is like this object here. The dimensions of it are about 1 to 2 micrometers. And so the volume of the E. coli is about 1 micrometer cubed. Okay, so we'll stop here for today. This is what I want you to have in mind, and we'll come back to it. The volume of the E. coli is about 1 micrometer cubed. Weight is picogram on average. If I take a typical protein made up of 100 amino acids, okay, the radius of the protein will be about 20 angstroms, if I think of it like a sphere. This is a unit that the biologists use for mass. We'll come back to it. And this is probably one of the very important things that we're going to revisit again and again is that the magnitude of forces at the nanoscale in biological systems is on the order of piconewtons. And it's fluctuations on the piconewton length force scale that drive the optimal ability of life, essentially. Okay, so that's where we're going to go on Thursday, before your exam. Okay, questions?
Are we going to finish all this outline?
I don't know, I doubt it. They're good. Yeah, now, well, it depends. So, if we make the class less interactive, then I can. So, it just depends. I mean, I don't care if I don't finish, actually. It's not so important. As long as you learn something new and, yeah. So, in all likelihood, we will not finish. Okay, see you Thursday.